last part of Chapter 17 review. We've already discussed transcription and translation. Now, how does this relate to mutations? A mutation is a permanent change in the DNA. So if the DNA is changed, then transcription is going to produce that same change in the RNA. Whether or not it affects the protein depends on the situation. So, let's say you have what's called a substitution. You substitute a letter for another letter. Well, what will that do? Well, it depends. For example, if UUU becomes a UUC, because this changed to a G, well, UUC still codes for the same amino acid. So that mutation will have no effect. Okay? So some mutations may not have an effect. Or suppose the substitution will change an amino acid. So for example, if you change this G to an A and this becomes a U, well, that's not arginine, it's going to be something else. So if you have an amino acid substitution, then the protein will be changed. How much depends on the situation. One example is sickle cell disease. Sickle cell hemoglobin only differs by one amino acid caused by one base pair point substitution. A point mutation is a mutation that occurs in one point on the DNA. So a base pair substitution is changing a letter for another letter. Another type of mutation is either an insertion or a deletion. An insertion adds a letter, and a deletion gets rid of one. So in either case, if you add a letter, well, what are we doing? We're going to shift all of these codons over. And every amino acid from that shift, that frame shift, is going to be different. So that could drastically affect the protein. In the case of functional proteins like enzymes, it will no longer be functional. A lot of recessive genes are non-functional genes. And that concludes Chapter 17.